Plastic. We use it, sometimes we reuse it, but ultimately too much of the plastic that we use is getting thrown away. How bad has this plastic epidemic gotten? In 2015, a study was done by Santa Barbara's National Center for Ecological Analysis and Synthesis, and it showed that 8 million metric tons of plastic are being added to our oceans every year. These plastics statistically kill 100,000 marine mammals annually, along with other birds and fish. And not only are these plastics harmful to the wildlife, but they're also detrimental to the ocean environments and ecosystems. So clearly, we have a problem. Let's take a look at a relatively new and possibly very effective solution to this very serious issue. Recently, Japanese scientists have discovered a bacterium that can actually eat a common type of plastic known as polyethylene terephthalate. Polyethylene terephthalate is a common plastic found in your regular everyday single-use water bottle. It's more commonly referred to as PET. In 2016, a team led by Dr. Kohei Oda from the Kyoto Institution of Technology and Dr. Kenji Miyamoto from Keio University collected 250 PET contaminated samples, including sediment, soil, and wastewater from a plastic bottle recycling site. They then took these samples and examined them, looking specifically at the microbes that were living on them to see if any of them were consuming PET as a source of growth. What originally appeared to be a handful of different species eating away at PET turned out to be one species of bacteria that was degrading it. They named this species Idonella saccharensis. After further research was conducted on Idonella saccharensis, they found that it secretes two enzymes that are responsible for breaking down these PET films. Once the bacterium adheres itself to the PET surface, it secretes the first enzyme. This first enzyme breaks PET down to an intermediate chemical called mono,2-hydroxyethyl terephthalic acid that Idonella saccharensis can then absorb. The bacteria then secretes the second enzyme, which breaks PET down even further, which provides the bacteria with carbon and growth. This is definitely a solid start, but there is the question of how fast can a community of Idonella saccharensis degrade polyethylene terephthalate. As it stands now, in an environment held consistently at 86 degrees Fahrenheit, a community of this bacteria can degrade a thin film of PET in about 6 weeks. This is some great progress compared to the couple of hundred or even thousands of years that would normally take a plastic to decompose. One issue with this is that it is highly unlikely for this process to happen in a natural manner. It's not very probable for a natural, highly polluted body of water to maintain a temperature for 6 weeks. So this process would most likely take a much longer time if it were to happen in a regular natural environment. Best use of this bacteria would come from specialized facilities where disregarded plastic wastes are brought and decomposed in communities of the Idonella saccharensis bacteria, kept under their own specific conditions that would allow them to be most effective. While this does provide great strides in reducing the plastic pollution epidemic our planet is currently undergoing, it is not an end-all be-all solution. If we continue to produce plastic waste at the same increasing rate that we are, this great discovery will be massively overshadowed. However, if we can manage to reduce the rate at which we are producing this plastic pollution, Idonella saccharensis could slowly but surely alleviate this plastic plague over time. Thinking about it realistically, it would just be better to recycle the plastic we use so it can be melted down to make new ones rather than allow it to build up and then later down the line need it to be managed. Another encouraging aspect to this discovery is that the enzyme itself has actually been slightly improved. Two separate groups who were doing research on Idonella saccharensis found that by subtly changing the enzyme's chemical properties so that it interacts with PET differently, made it work more quickly than the natural PETase. Although these changes were relatively small, it's still significant to see that the enzyme can and has been improved. Since it is difficult to modify an enzyme to work better than it naturally would, the fact that scientists were able to improve could possibly mean it is a newly evolved bacteria and that it shows even more room for growth and improvement. Another thing to consider is that we do not yet know for sure how common this process is worldwide. Now that we know what kind of bacteria that we're actually looking for, we might be able to find it in places all over the globe. This would allow for more use of the bacteria and more research done on the bacteria. We have dug ourselves into a pretty big hole when it comes to plastic pollution. Even as we stand now, our plastic input is estimated to double by the year 2025. There's a lot to be done, but it's good that we can see progress towards being able to lessen our plastic input on the planet.